So these are the two mechanisms. These were the two mechanisms which exist to ensure that the stem cell population is maintained. So self-renewal is one of the most important attribute of any stem cell. The second one was potency. So then we also categorize the cells or the different type of stem cells on the basis of their potency. Now, when I said potency, as I said, they are undifferentiated cells under certain physiological or experimental condition. They can be induced to become a cells with a very specialized function. Like if you want to make a neuronal cells, you can make a neuronal cells from the stem cells. If you want to make a cardiac cells, you can make a cardiac cells from undifferentiated stem cells, provided that you should, or I would say that you should provide the milieu or conditions which would be convenient for a, or which congenial for a lineage, um, for a specific lineage which you want to make. For example, if you want to make a beta cells from the stem cells, then you have to provide a very specific culture conditions in which you can navigate your cells toward the beta cells. So if we have to categorize the stem cells on the basis of the pluripotency, on the basis of potency, you can categorize them as a totipotent, pluripotent, multipotent, and the unipotent. Then we also discussed that what is the totipotent cells. I gave you the example that like identical monozygote twins would be an example of a totipotent stem cells, because monozygotic twins are coming from a same embryos. Like at the two cell stage embryos, there are two blastomeres, and each blastomere has the potential to give rise to an entire organism on its own. So two cell and the four cell stage embryos, uh, all the blastomeres which are present in the two cell and the four cell stage embryos can be considered as a totipotent cells, because they have the ability to give rise to an entire organism on their own. And we also discuss about the pluripotent cells. As I said, that pluripotency means they can give rise to all type of cells. Like we have 200 type of cells in our system, in our body, and they have the capacity to give rise to all type of the cells. But they cannot give rise to an entire organism on their own. So that difference between the totipotent and pluripotent stem cells would be that totipotent means they have the potential to give rise to an entire organism on their own, while pluripotent stem cells, they can give rise to all type of the cells, but they cannot give rise to an entire organism on their own. They have to take the help of a trophectodermal cells to give rise to an entire organism. Then we also discuss about this multipotent stem cells, like we will to here multipotent and the unipotent stem cells. So I gave you an example of cord blood stem cells, mesenchymal stem cells. They are a typical example of the multipotent stem cells. That means they can form all type of the cells, or not all type of the cells, but a few type of the cells. They cannot form all type of the cells, while the unipotent stem cells are like the progenitor stem cells. So they can give rise to cells of their own. That's what we already discussed. Then after that, I also taught you that how you can drive embryonic stem cells, as the name suggests, that all the embryos have the inner cell mass. It means the first differentiation takes place at the blastocyst stage, and you get trophectodermal cells and the inner cell mass. If you can capture the inner cell mass, and you can try to culture in a culture system, which is congenial for the embryonic stem cells, then you would be able to capture the embryonic stem cells. So in case of the human, you can take a day 5 blastocyst, then you can isolate the inner cell mass by using immunosurgery or the mechanical method, and then you would be able to capture the ICM cells, and you will get a embryonic stem cells from the ICM cells. That's how we drive the embryonic stem cells from the IVF embryo. Whether it's a human embryos, whether it's a mouse embryos, whether it's a livestock embryos, if you can generate the embryos from the in vitro fertilization method, you can grow them till the blastocyst stage, then you can isolate the ICM cells, and you will be able to capture the embryonic stem cells. So then we also compared the human embryonic stem cells with the mouse embryonic stem cells. So morphologically, they look a bit different, like, but both grow in the form of a discrete colonies. If you will see here, and the mouse, the human embryonic stem cells they grow in the form of a discrete colonies. And if you will see it properly, I would say if you will check every individual cell, then you will see the nucleus is very much dominant and they have a high nucleoside concentration. And and it's pretty much same with the mouse embryonic stem cells. The difference would be these colonies are with dome shaped. In case of a mouse embryonic stem cells, the colony is with dome shaped, while in case of a human embryonic stem cells, the colonies are with flat as compared to the mouse embryonic stem cells. Then we also discussed that, okay, what is the use of this embryonic stem cells? Like you can generate the embryonic stem cells from any random blastocyst, and then you can navigate or differentiate them in the type of cells you're looking for. I give you an example of a diabetes. Like in case of a diabetes, the it's autoimmune disorder, we use beta cells. So you can replenish beta cells by navigating the embryonic stem cells toward beta cells. But again, the problem would be, the issue would be the immune rejection, because these cells are coming from a very random embryos. So the chances are that when the moment you will transplant back into the patient, it is not going to accepted by the body of a patient. It will be rejected. So embryonic stem cells definitely for the research purpose it's important, but as far as using them for a regenerative medicine or for therapy, I would say it's a big no. And the second thing is ethically, people have a lot of issues with this, especially in the European countries. We believe the moment we use or destroy the embryo, we destroy the one life. So in few countries, the research on the stem cell, embryonic stem cells is also banned. Then the other option was therapeutic cloning. So I guess uh, technically it's doable. You can take a cell from a patient, they can grow a cell line, then you can use this cell as a donor cell, and then you can transfer these cells back into the enucleated oocyte. Then you can grow them as a blastocyst. Then you can isolate the initial mass, and then you can capture the embryonic stem cells. Now we want to generate the beta cells, so any type of cells you're looking for, you can generate and then you can transfer them back into the patient. So technically it's doable, I would say theoretically it's doable, but it's very difficult to clone the human embryos. There are a lot of issues because the number of oocytes which are available is very less. Second thing is availability of the oocytes. So there was a scientist in the Korea whose name was Wang that he claimed that he cloned 11 cell lines, but eventually it turned out to be fraudulent, and then we left with no other option. Then there was a scientist from Japan. His name was Shinya Yamanaka, and he came up with the concept of like induced pluripotent stem cells. As I said, that induced pluripotent stem cells means that you can take a cell, a somatic cell, you can induce a pluripotency by the ectopic expression of certain transcription factors like OX4, OX2, NOK, NF4, all these are transcription factors. So you can put them in these cells and eventually you would be able to generate the induced pluripotent stem cells, which are very much similar to the embryonic stem cells. So that's what the Yamanaka did along with his postdoc Takahashi, and they replicated the same technology in the human with the human cells, and he was able to generate the human Eyepieces and as well as the mouse Eyepieces by using the same method. Now, then we discussed about the garden experiment as well yesterday. Uh, so Yamanaka got this clue from this garden experiment where he cloned the tadpoles. So he took the epithelial cell of the frog, then he enucleated the oocytes of the frog, and he transferred that donor cell back into the enucleated oocyte, and then he transferred that embryo back into the surrogate, and he was able to get the tadpole. So, but he never got the frogs. That's a different story. But the concept of this iPSC came from this experiment. So this experiment provided the key that somatic cell can be reprogrammed back into the embryonic state. So it means there are certain factors in the oocyte which can reprogram the terminally differentiated cell back into the embryonic state. Now, I also showed you this list of these factors in the cells. So, like Yamanaka gave the concept of the iPSCs way back in 2007, and I would say it's been almost 14 to 15 years. So mouse and even iPSC cells have been generated in a variety of ways. And if you will see here, the most of the strategies are DNA based or the virus based. However, there are one or two exceptions as well, like protein based strategies. You will see recombinant proteins people have used. But in the protein based strategies, the recombinant protein which is being used are very, very challenging to generate. And if you find the quantities required, it's a tough job. So the clinical applications of the Ipscs is limited by the fact that most of the protocol modified the genome to affect the cellular reprogramming because it carry the risk of ancestral mutagenesis. If there's a DNA, there's a virus, then I would say that Ipscs which you will generate won't be of clinical grade. So if you want to use iPSC for a therapy, then you have to develop a method by which you can generate a clinical grade iPSCs. So there are few options which are very much there, which can be used to generate the iPSC that they use to reproduce themselves. So one of the method is like a non-integrating strategy. So what people have done actually, so they have used a non-integrating strategy by the administration of the synthetic mRNA molecule. So they modified the mRNA molecule to avoid any apoptotic response against the mRNA molecule. So if you are using an mRNA molecule, so it means there is no need to perform your experiment in a stringent biological containment. Second thing, no DNA means completely eliminates the risk of genomic integration or ancestral mutagenesis. And whatever the iPSCs you will generate by using this non-integrating strategy, that is definitely going to be a clinically free. So if you will use mRNA molecule or the cocktail of the mRNA molecules of course of nano, KLF4, RAIN20, or the transcription factors, then the chances of having the ancestral mutagenesis will be completely eliminated. It means this strategy is a safe strategy to generate a footprint-free induced pluripotent stemcells, which are going to give a clinical grade. Now we also discussed yesterday about this timeline. So take-home message from this timeline. This timeline is to generate the iPSCs by using a mRNA molecule. So I'm giving an example actually how you can generate an iPSC by using this mRNA molecule, like the messenger RNA molecule. This is a synthetic mRNA molecule which is being generated by the in vitro transcription. Now, if you will see this timeline, the take-home message from this timeline will be that repetitive transcription regimen will be required to sustain the high level ectopic expression of these protein molecules. Here you can see from day 1 to day 15, what people are doing actually, they have transcribed the cells or the target cells every single day. So for the transcription, you can use the RNA max reason the different, and you can use the lipofectomy as well. You can also use the electroporation method so that you can get your RNA inside the cells. So starting point is very simple. We can start with the different intensities of the cells, like target cells could be 1000 cells, 1500 cells, 5000 target cells. So when I say target cells, it means that these are the cells which you want to reprogram in the iPS cells. So when you so target cells are nothing, they are the even fibroblast cells. Now you have taken them as a candidate which need to be converted into a induced pluripotent stem cells. So when you start, you don't know which density is going to work or how many cells we should take to reprogram. So what the researchers have done, they have taken a different actually density for the target cells, and eventually they wanted to know which density is I would say optimum to generate the iPSCs. So they came up with the idea that let's start with 1000, 2500 and 5000 cells. And they seeded these target cells on, you can see it on the murine embryonic fibroblast cells. They seeded this on a newborn posting fibroblast cells. So why they treated these cells on a NEFF or maybe a murine embryonic fibroblast cells? Because they need some substrate. In this case, the people have used cells as a substrate, but you have to inactivate these cells. Otherwise, these cells will supersede the growth of your target cells. So you can see here, I mean, these are the newborn posting fibroblasts, which we have inactivated by using the micromycin C. So once you believe that whatever the cells you are using as the fetus cells are very much inactivated, then you can see the target cells survive. And then you can start the transfecting the cells. But before the initiation of the first transfection, what you need to do, you have to treat your cells with a B18R molecules to avoid any apoptotic response against the this foreign mRNA molecules. So B18R is nothing but it's a class 1 interferon inhibitor. So after the transfection, when you start a transfection, day 1, day 2, day 3, day 4, day 5, day 6. On day 6, the morphological changes were very much evident. You can see these cells are looking very different from the fibroblast cells. So after day 6 of the transfection, the morphological changes were very much evident. Now by day 11, you will see that the morphological changes were more evident. So if you will have a close look here actually, and 10x magnification, you will see these cell have a high nucleocytoplasmic ratios as well. You see here, even on day 11 at 10x, you can see that the cells have transformed into a iPSC-like cells, and even you can see obvious boundaries here because, as I said, they grow in the form of a discrete colonies. So you can see the morphological changes, and you will see they've started migrating actually once they are transformed into an Eyepieces, and you will see some colonies are popping up here on day 11. By day 18, you will see these colonies are very much humongous, very large in size. You can see if you will closely interrogate these cells individually in the discrete colonies, you will see high nucleocytoplasmic ratio. So by day 20, actually, because you kept on transfecting the cells every single day, by day 10, it was the morphological changes were very much evident. And on the basis of morphology and then a stringent pluripotency marker, that is the tumor rejection antigen 181, we picked the subset of 12 clones because there is every chance that you might end up with some pseudo clones as well. So on the basis of morphology and the stringent pluripotency marker, we picked subset of 12 clones, then which were cultured further in the standard embryonic stem cell media. So whether it's a mouse embryonic stem cells or the mouse iPSs, or whether it's a human embryonic stem cell or human iPSs, they need to be cultured in a very different culture. For example, like in case of a mouse iPSs or the mouse embryonic stem cells, we have to add the leukemia inhibitor factors. While in case of a humans, actually, we don't add leukemia inhibitor factor. Instead of a leukemia inhibitor factor, what we do, we add the FGF system. It means to keep these cells, whether it's a mouse embryonic stem cells or the human iPS cells or any pluripotent stem cells, the signaling pathway is different. So they follow a different pathway to keep these cells in an undifferentiated state. So as a reference point, what you can see here, you can see these different signaling pathways here. So we have compared the mouse and the human like iPSCs or embryonic stem cells. What kind of a signaling pathways they follow to keep themselves in an undifferentiated state. So it's very much established that mouse serum, like a serum, is the best proxy for this BMP, that is a bone morphogenetic protein. So serum and the leukemia inhibitory factors is required for the pluripotency in the murine stem cells in vitro conditions. So one is serum, second one is murine leukemia inhibitory factors is required to maintain the cells or maintenance for the maintenance of the pluripotency in the murine. You need LIF and serum. So what's happening here, if you will see here, that leukemia inhibitory factor is there. So leukemia inhibitory factor, what it does, it is going to bind to the leukemia inhibitory factor receptor. It's very much at the cell surface. Now, this is going to form a heterodimer with the GP130, that is a signal transducing unit, and this will lead to the activation of a JAK-STAT pathway. So the bottom line is, the moment the lift bind to the leukemia inhibitory factor receptor, it activates the STAT3 or activation of a JAK-STAT3 takes place. Along with the JAK-STAT pathway, you will also notice that mitogen activated protein kinase cascade is also got activated. So STAT3 and the MAP kinase pathway is being activated. So once this STAT3 is activated, it is going to get phosphorylated and it will form a dimer. And before being translocated inside that to the nucleus, we are going to act as a transcription factor. So it will be phosphorylated and it will form a dimer before being translocated inside the nucleus. That's how they maintain their pluripotency. But what I said, along with this leukemia inhibitory factor, you also need serum, because serum is a cheap proxy for the BMP4. It means the key factor which control this process of pluripotency is BMP. So when you culture mouse embryonic stem cells or the mouse iPS cells, you have to add the leukemia inhibitory factor and you have to add the serum to your culture measurement as well, because serum is the best proxy for the bone morphogenetic protein. And the downstream target for this STAT3 is c-Myc. So that's what we have learned, that when you culture the mouse embryonic stem cells, mouse iPS cells, or I would say mouse pluripotent stem cells, you have to add the leukemia inhibitory factor and the serum in your culture system. Now, as far as human iPSCs or human pluripotent stem cells are concerned, for the maintenance of the human embryonic stem cell or human iPS cells in an undifferentiated state, you don't need leukemia inhibitory factor at all. So the lift is not required for culturing and maintaining of the human embryonic stem cells. However, lift phosphorylates STAT3 in the primate embryonic stem cells, as in case of the mouse embryonic stem cells. In case of a human pluripotent stem cells, what you can see that TGF-beta signaling play a very important role. And it play a very important role in the maintaining of pluripotency and in self-regulation. So they signal via this SMAD1 and SMAD1 and activate nodal pathways, you can see here. And so the point is, what I'm trying to tell you says that if you will compare the mouse embryonic stem cells and the mouse pluripotent and the human pluripotent stem cell, the signaling pathways very, very different to keep the cells in an undifferentiated state. So TGF beta is very important. Apart from that, FGF is very important. What I said, you also add FGF to your culture system to maintain the human embryonic stem cells YPSs in an undifferentiated state. So FGF means fibroblast growth factor 2 is very much proficient in maintaining human pluripotent stem cells without serum and fetal cells, because serum is a cheap proxy for the bone morphogenetic protein. So if you will add the BMP to the human pluripotent stem cell culture system, it will differentiate your cells. So what FGF2 does, FGF2 promote the self-renewal of the human pluripotent stem cells by activating the PI3K pathway. It's very simple. And this leukemia inhibitory factor, it also acts downstream of the phosphorylated pathway. So what I'm trying to tell you that two pluripotent stem cells coming from a two different species, they have a different signaling pathways to keep themselves in a non-differentiated state. It means culture system is very different. So you cannot use the human pluripotent stem cell culture system for culturing of the mouse pluripotent stem cell. And And the culture system which we are using for a mouse pluripotent stem cells cannot be used for culturing on the human pluripotent stem cells. Now we know the culture system is different. So once you will get the iPSC or the pluripotent stem cell in your hand, you can use a standard human embryonic stem cell medium. And after that, you have to characterize them as well, because whatever you got in your hand, how would you know that they are bona fide pluripotent stem cells? So you have to assess their pluripotence. So you can do it at RNA level as well. So you can isolate the RNA, convert them to cDNA, run on gel, and you can check the expression of these factors. If you see that they are showing some positive expression, then you can see whatever we got are pretty much similar to the pluripotent stem cells. So once you are done at the mRNA level, then you have to check at the protein level as well, that whether these factors are able to meet protein or not. So you can check versus the pluripotency at the protein level. So you will see that the cells which we generated by using this non-integrating approach, by using the cocktail of the mRNA molecule for showing the expression of phosphorus, oxygen, and all, SSCA4 disease state specific embryonic antigen, tumor rejection antigen 1, tumor rejection antigen 1. So they were very much positive for all these stringent markers for the human pluripotent. So they were also positive for the alkaline phosphatase staining. So once you got the cell lines, you can see these are the different colonies. These are the colonies of the human pluripotent stem cells. So what you can do, you can use alkaline phosphatase staining to see whether your cells are positive for this alkaline phosphatase or not. So eventually, by using this method, what you can do, you can get this iPS cell line, that is your human induced pluripotent cell line. You can maintain these cell lines on some feeder layer, like you can inactivate neurine chronic fibroblasts, then you can massage your cells on these feeder layers. Otherwise, there's new culture conditions also there which are feeder free. So you can seed your cells on a matrix gel, and you can use a commercially available empty cell medium. So in that case, you don't have to use a feeder cells to obtain the culture. So what we have learned that signaling pathways which maintain these cells and kids from mouse and the humans are very much different. Like in one case we have to use the leukemia inhibitor factor, in another case we have to use the FGF. In case of a human embryonic stem cell, the pluripotent stem cell culture, we don't use serum, because as I said, serum is the best proxy for the BMP. If you will add the BMP to your culture system, it will differentiate your cells. Now at this stage, you can say that you got some cell lines, that is your pluripotent stem cell lines. Now at mRNA level, they have shown some stringent markers. At the protein level, they have also shown some markers. They were positive for certain markers. But these are not the gold standard assays. So the gold standard assay is that what I said, I will tell you that by definition, stem cells are undifferentiated cells, and under certain physiological or experimental condition, you can make the type of cells you're looking for. It means they should have a potential to give rise to all three types of germ layers, that is ectoderm, mesoderm, endoderm. Unless they form these three germ layers, or they show that they can form these three germ layers, I cannot consider them as a bona fide pluripotent stem cells. So to assess the pluripotency, it's very important to subject these cells for some functional assays. So what are these functional assays? So functional assays involve the in vitro and in vivo differentiation of these Ipscs or the induced pluripotent. Now, what are those functional assays? So the one function assay is in vivo differentiation. What does it mean in vivo differentiation? So what you can do, you can take your induced pluripotent stem cells, that is your Ipscs, whatever you have generated. So these Ipscs, the human Ipscs, they grow in the form of a discrete colonies. Now, there is a difference in the mouse embryonic stem cells or iPS in the human iPSCs. So human iPSCs, if you will try to make a single cell suspension by using a collagenase, then there is every chance that they might get differentiated. So the easiest way would be that you have to actually you can use some displays and the collagenase, and then you cannot be harsh on your human iPSC cells. So along with these cells, you can add matrigin or add matrigin, and then you can inject at almost 1 million to 2 million cells in a SCID mice. So what you have done, because you're trying to perform you are trying to assess a pluripotency by performing some functional assays. One functional assay is what? In vivo differentiation. So you've generated iPSCs in in vitro conditions, and you want to check whether they have the potential to form all three germs here or not, that is ectoderm, mesoderm, and endoderm. For that, what you are doing, you are putting these cells, you have made a single cell suspension, you've added a matrigin, and some TBS, and then you've injected in a SCID mice, that is an immune compromised mice, so that there won't be any chance of immune rejection of this. Because cells is of human origin and mice is mild. If you will use a normal mice and you will inject your human iPSC, then there's every chance of immune rejection. So to make sure that there won't be any immune rejection, what to do? Use a SCID mice or the immune compromised mice. Now, what you need to do, you need to inject your cells in the mice, maybe under the kidney capsule, and after 4 to 5 weeks, if you will see the formation of a teratoma, then you can say that okay, fine, my cells, whatever the cells have generated, are pluripotent stem cells. So you can take out the teratomas and do the sectioning, and then you can look for ectoderm, mesoderm, and endoderm. This is the in vivo differentiation of your cells. It means you have tried to assess the pluripotency of the cells which you are generated. Now the second option is to assess the pluripotency is your in vitro differentiation. Now for the in vitro differentiation, what you can do, because these two are functionals, like whether it's in vitro differentiation or in vivo differentiation, the bottom line is we are trying to see whether they can form all three germ layers, ectoderm, mesoderm, and endoderm. So in vivo, inject the cells in an animal, wait for 4 to 5 weeks, if they form the teratomas, isolate the teratomas. If you see all the three germs layers, then it means definitely they have to be structures. Now for in vitro differentiation, if you don't have the facility of this SCID mice, so what you can do, you can try to generate some embryonic bodies. Now, how you generate the embryonic bodies? So, if you are maintaining your human embryonic stem cells, the iPSCs in a culture system, for that, what you do, you add fibroblast growth factor, you put them on a feeder layer, you use serum replacer, you don't serum at all, so for that, you need serum replacer, and you provide them a culture condition in which these cells can remain in an undifferentiated state. Now you want to make an invite bodies from these cells. Why we are trying to make an invite body? Because idea is to assess the pluripotency of these cells. So why we are trying to assess the pluripotency? Because we have generated by using a non-integrating strategy. We have used one strategy to generate the Eyepieces. We pick some clones. Now there's every chance that you might pick some wrong clones as well. But you have to check whatever you generated are bona fide pluripotent stem cell. So this in vitro differentiation method means that idea is to generate the invite bodies. So for that, what you need to do, you need to withdraw all the factors which are keeping these cells in an undifferentiated state. Like you have to withdraw the feeder layer, you have to withdraw the fibroblast growth factor as well, to make sure that they should start differentiating. But if you will keep these cells or these colonies or these cells in another culture dish, then they will again attach to that culture. You won't be able to get the infant models. So for that, what we do, we isolate what we will do, we are going to cut these colonies into few small fragments by the mechanical method. We will pick it mechanically and we are going to put them in a suspension. In a suspension, we are not going to add any FGF or any other factor which will keep them in an undifferentiated state. So that's how you get the embryonic body. The embryonic body is what? They are the aggregate of the cells which are coming directly from the pluripotent system, right? So these cells are pretty much derived from the pluripotent structures. So they will aggregate and differentiation will be initiated and they will try to recapitulate the embryonic terrain. And after that, what we can do? Again, you can just do the sectioning on these embryonic bodies. And then you can check whether they can form this ectoderm, mesoderm, or endoderm or not. So in both functional assays, whether it's an in vitro differentiation or there's an in vivo differentiation, so idea is very simple. We have to look for the all three germs here, ectoderm, mesoderm, and endoderm. Like mesenchymal stem cells. Mesenchymal stem cells are considered to be a stem cells, but there's second school of thought as well. People believe that they are not stem cells; they are stromal cells. Why? Because if you will try to generate the neuronal cells from mesenchymal stem cells, you can generate neuronal cells. But again, that would be functional or not, it's a big question mark. So that's why it's very important to assess the pluripotency or the potency of the cells which you have generated. So these two are the functional assays. But again, even after getting the results from these assays, possible results, you cannot say that you got a bona fide pluripotent structures. For that, you need to test it or you need to subject these cells for more stringent assays. And the most stringent assay, or I would say the gold standard assay, would be the chimera formation. So we'll go into the chimera part as well, but before that, I will tell you schematically how you can form the simplified bodies. So what we did, we took the Eyepieces that is induced pluripotent stages, then we cut them mechanically into the small fragments, and then we pull them in a culture dish or not a culture dish, ultra low attachment dishes. And we withdraw all the factors for keeping them in an undifferentiated state, right? And then we did the sectioning of this embryonic bodies. Idea was to figure out whether they can form the all three germs there. The second one was the in vivo differentiation. You can inject your cells in the mice, and then you can wait for a few weeks to see whether they can form teratomas or not. So if you can narrow it down, this experiment. So the idea was to see or assess the pluripotency. And how we are assessing the pluripotency? By checking by whether they can form all the three germs they have. So there are different ways by which you can generate the embryonic models. So if I have to show you all different ways, like one way is this: you can have your cells, put them in an ultra low attachment edge. We draw all the factors which are keeping them in an undifferentiated state, and then you can get your embryonic bodies. Another method is hanging drop method, and then microwave method. So I will tell you a bit about this hanging drop method, how you can do this hanging drop method. So if you will take an embryonic stem cell IVF, this induced pluripotent stem cells maybe from mouse, from human. So this is from mice. This protocol is from the mice induced pluripotent stem cell for the mice embryonic stem cells. So now your cells are growing very much in a congenial environment. What you have done, you've added the leukemia inhibitor factors. You have added a feeder layer, so you have treated your cells on a feeder layer. You have added a leukemia inhibitor factor and serum. So you are providing a very cozy environment for these cells. Now they will remain in an undifferentiated state. They are not going to differentiate. Now what you can do, you can take out these colonies, maybe you can add to these colonies, and you can make a single cell. Because what I said, if you remember, I told you that these embryonic stem cells are cells, they grow in the form of a discrete colonies. So within the colonies, you will see the thousands of cells are there. So what you need to do, you need to subject these cells for the trypsinization so that you can get a single cells. Once you will get the single cells, what you can do, you can make a droplet, maybe a 20 microliter droplet on the lid of a culture dish, a 20 microliter droplet of a embryonic stem cell medium without leukemia factors. So you can add almost 300 to 500 cells in each droplet. So the size of the droplet might vary from 20 to 30 microliter. And the thumb rule is there is no leukemia inhibitor factor at all. So after 5 to 6 days in the honey drop culture, you can plate this embryonic bodies which you got after 5 days into a culture dish or maybe a children culture dish, and then they will definitely get attached, and then they will start differentiating more towards a lineage in which you are interested. So in this case, what these people have done actually, they have used a very specific method or a culture medium which will navigate these cells toward the beta cells. So they have used a dikaiosome kind of the culture system for the medium, so that they can get the beta cells. So after some time on the day 21, what they did actually, they did the staining with the dikaiosome on day 21 and day 28 to analyze whether they got the or to analyze the pancreatic islet fluid gene expression and insulin immunoreactivity on day 21. So this is how you can generate the embryonic bodies. This method is known as hanging block method. So this method is being used very people use this method frequently for the generation of mouse embryonic bodies and then mouse embryonic bodies from the mouse Eyepieces of the mouse pluripotent stem cells. In case of the humans, you can also do the same thing. You can use a hanging block method to use to generate the embryonic bodies. Or if you are not comfortable with this hanging block method, what you can do, you can put them in a ultra low attached mesh, then you have to withdraw all the factors which are keeping these continent undifferentiated. And after 5 6 days, you can take out these cells of the embryonic bodies and receive them on a maybe on a substrate so that they can start differentiating. So what I said, there are different methods by which you can assess the pluripotency of the Ipscs which you have generated. Or even in case of embryonic stem cell, when you generate the embryonic stem cells, you have to assess the pluripotency. So there are different methods. You can do it at mRNA level, you can do it at protein level by just seeing the expression of the stringent markers. But you have to perform the functional assessment. Functional assessment in vitro and in vivo differentiation. So in vivo differentiation means you have to inject your cells in a stag mice of the immune compromised mice, and if you get the teratomas, you can say that okay, fine, that and then you have to do the section to see whether all the three term spheres are there or not, because we have to stick with the definition of the theory. And definition suggests that your cell should have a potential to give rise to all three terms. So that's what we are trying to test, that whether the Ips which we have generated have the potential to give rise to all type of cells. Because technically you cannot try to differentiate individually one by one from neuron to all muscle cells and all and 200 type of cells. The easiest way would be if you can see that your cells have a potential to form ectoderm, mesoderm, and endoderm, and definitely you can get all 200 type of cells which exist in your body. Now, apart from that, there is one more method to assess the pluripotency, which is known as tetraploid complementation assay. What is this tetraploid complementation assay? This is also one of the functional assays which can be used to see whether your pluripotent stem cells or the Ipsc which you have generated are bona fide Ipscs or bona fide pluripotent stem cells. So it's very simple. In the tetraploid complementation assays, what you need to do, you have to take two embryos. And what does it mean? So you will take two cell stage embryos. Two cell stage embryos. So if you will take two cell stage embryos, if you will fuse them, what you will get? You will get a tetraploid embryo. What you will get? with the tetraploid embryos. Now these tetraploid embryos can be cultured further and then they will give rise to a blastocyst. But if you will transfer the blastocyst into the surrogate, then you won't be able to get an organism. So now the idea is, can we use this assay to check whether our cells which we have generated are pluripotent or not? What you can do, you can have your Eyepieces, you can tag your Eyepieces with the GFP, and then you can inject your cells, whatever you have generated, into a tetraploid blastocyst. And then you have to transfer the embryo to the recipient. Now, the point is, what the point I'm trying to make, that if your pluripotent stem cells are bona fide pluripotent stem cells, it means they would be able to rescue these tetraploid embryos. Why? Because these tetraploid embryos are not going to give rise to an entire organism. But what you are doing in this essay, you are using the trofectodermal cells of this tetraploid embryos. So whatever the cells you use, they are going to contribute to the formation of an organism. And you are using the trofectodermal cells of this blastocyst, which is coming from the tetraploid embryo. So it means at the end of the day, if your pluripotent stem cells or the iPS cells which you have generated can rescue the tetraploid embryo, it means that whatever the cells you have generated are the bona fide pluripotent stem cells. So there are different methods by which you can assess the pluripotency. But having said all these things, what I said that the gold standard assays would be your chimera formation. So you might have seen these chimeras. The chimeras means any organism, a single organism which is composed of the cells with a distinct genotype can be considered as a chimeras. So what you can do, a very simple method is there. Then you can have your cells, you can inject your cells, your Eyepieces into the blastocyst of another mice. Like there are two mice of different strains, maybe of different colors. So you can generate the Eyepieces from one mice, and then you can generate the blastocyst from the other mice, and then you can transfer your iPSC into the blastocyst of another mouse. If your cells, which happen with pluripotent cells, if they are bona fide, then definitely they will get mixed with the item cells of the blastocyst, and then they will contribute to the formation of an organ. Here you can see that this guy is having the genotype or the composed of the cells with two distinct genotypes. And because in case of a human, it's unethical, so you have to perform this experiment in the mice. So I guess in humans, ethically, you cannot do it. So you won't be able to figure out that the cells you have generated are bona fide pluripotent stem cells or not, because you're not able to generate the chimeras, because you're not allowed to generate the chimeras in case of humans. But on the basis of your other functional assays like tetraploid complementation assay, in vitro differentiation, in vivo differentiation, you can at least say that you have generated cells very much close to the embryonic stem cells, or very much similar to the embryonic stem cells. Now, the point is, I will give an example that how you can navigate these Eyepieces toward a certain year. So once you will perform all these assays, and after performing all these assays, you can see that you have generated a bona fide cell lines. Then you can do a karyotyping as well to see the chromosomal profile of the cells. If they are very normal cells, no chromosomal abnormalities found, then what you can do, you can navigate them toward the cells you are looking for. Suppose somebody is suffering from a myocardial infarction. Myocardial infarction means what? I mean, it's like a cardiac, maybe heart attack. If it's a major heart attack, the patient loses quite a few cardiac cells. But the problem is these cardiac cells will be replaced by the fibroblast cells. So the functionality of the heart will go down. So as a stem cell biologist, what we can do, we can try to replenish or restore the lost cells. How you can do that? You can take the cell from the patient. You can induce the pluripotency in the patient, and then you can try to navigate this cells towards the cardiac cells. Once you will generate the cardiac cells, then what you can do, you can transplant back into the patient to restore the functionality. So I will give an example how you can do this. So cardiac differentiation, if you will see that you have this human pluripotent stem cells. So if you want to generate the mature cardiomyocyte, you have to navigate these cells toward the mature cardiomyocyte. So it means there are different signaling pathways involved. But for me, when I see this slide, I would say the wet signaling pathway seems to me the most important pathway. At one stage here, you can see the upregulation of this wind signaling pathway. At one stage, there's a down regulation of the wind signaling pathway. So it means there's a tight regulation of the wind signaling pathway. So you can optimize the protocol to generate the cardiomyocytes from your HPSC. So you can use these factors or the biomolecules at the different stages to get the mature cardiomyocytes. Now, this is what we developed, this protocol we developed in the lab. The idea was very simple. On day zero, once you got your human pluripotent stem cells, on day zero, you CHIR, that this is your G-STIP3 inhibitors, and then it actually activate the wind signaling pathway. And then day three, you can use the wind signaling pathway inhibitors, and then on by day 40, you will get what? The contracting cells. What I'm trying to tell you that if you have your pluripotent stem cells, a bona fide pluripotent stem cells in your hand, you can navigate them to other type of cells you are looking for. If you want epithelial cells, you can generate. If you want cardiac cells, neuronal cells, intestinal cells, you can generate by navigating these cells towards the kind of cells you're looking for. Having said all these things, you have to have a knowledge of the signaling pathways. Then only you would be able to manipulate or maneuver all the signaling pathways to get the cells. So in our case, we knew that this wind signaling pathway is very much important in the regulation or in the formation of the cardiac cells. So we optimize and we realize that just by upregulating and downregulating at two different time points, you can get the contracting cells. So let me show you what we got. So let me see if this works or not. This is a video of this contracting cells, which is not working anyway. So these are some see if it is working. Yeah, you can see. So what we generated cardiac cells by using this even pluripotent stem cells. So let me show this one. Okay, you can see these cells are beating, right? Okay. So the idea was that you can take bona fide pluripotent stem cells. You can use a signaling pathway, but you should have the idea how the signaling pathway works in case of a pluripotent, in case of cardiac cells. So then what we did actually, because the biomolecules are very expensive, so for that what we need to do, you need to narrow it down to the very specific important biomolecules which can be used to generate the cardiac cells. So with just by upregulating and downregulating the wind signaling pathways, we were able to get the beating cells. Now, once you get these beating cells, what you can do, you can do some immunohistochemistry analysis as well. Like you can see whether they express dopamine T, imagine heavy changes there or not, imagine light changes there or not, alpha actinin is there or not. So you can do immunohistochemistry as well to see whether your cells, whatever you have generated, show the expression of these markers or not. So you can do some immunofluorescence images as well. You can dissociate your contracting red body cells, and then you can look for the different markers like NKX2.5, MSCA2, CRCA2. So there are different kinds of oxidation markers which you can use to check whether you got a bona fide your cardiac cells or not. Now, the one important aspect of any cardiac cell is that they beat. Now, they beat because they have channels, they have potassium channels, they have sodium channels. So, we wanted to determine whether this human pluripotent stem cells, which we have used to deliver cardiomyocytes, can display functional electrophysiological properties on that. So what we did actually for that, we micro dissected the beating area, like I showed you few pictures where the two part, a small part of that particular cell was beating. So we micro dissected that beating area, and then we plated it on the micro electrode and it plates. And then we started monitoring for the beating abilities, each beat at a minute. So under the microscope for 72 hours period, we monitored the beating abilities, and then we also checked the effect of the different drugs on the electrophysiological properties of cardiomyocyte from the human pluripotent cells. And we also evaluated the ability of these myocytes to respond to the pharmacological stimuli or not. So eventually, our cells were able to show the apt response to the chondriotropic stimulation or chronotropic stimulation. So once it means what? So why I'm trying to give you this example? The example means that if you have the pluripotent stem cells in your hand, you can generate a different kind of cells in which you're interested. So we were interested in the cardiomyocytes, so we generated the cardiomyocytes. As simple as that. So if I have to summarize whatever I said, you can take a fibroblast cells, you can generate the Ipscs. So how we generated the Ipscs, we transfected the cells with the mRNA molecules, the cocktail of the mRNA molecules, and we were able to get the induced pluripotent stem cells. Now, once you will get the induced pluripotent stem cells, then you have to characterize these cells at a different level. So we characterize them at the protein level as well as at the mRNA level. Then we also perform certain functional assays like in vitro differentiation, in vivo differentiation, tetraploid complementation assays, as well as you can go with a chimeras formation if you have this mouse embryonic stem cells. And we also try to generate the cardiomyocyte, but nowadays we are trying to generate the beta cells, whether our cells can form the beta cells or not. So this is the summary of the Eyepieces. So if I have to summarize the Eyepieces which we have generated, they are like P4 predictive, preventive, personalized, participative. Predictive means safety and efficacy. Preventive means adverse drug reaction or disease. Personalized 100% personal to you. Participatory definitely individual donate the samples, and then you can use your own cells for the cell therapy. So this is all about the Ipscs, but induced pluripotent stem cells are pluripotent stem cells, that's for sure. But the story starts way back from 1981. The first person who isolated the embryonic stem cell from the ICM cells of the blastocyst in the mouse was Sir Martin Evans. Then in 1998, Jamie Thompson, he isolated the ICM cell from the human blastocyst for the first time, and he was able to generate the human embryonic stem cells. And then eventually in 2007, Yamanaka and Takahashi used the um, transcription factors to generate the induced pluripotent stem cells. So I guess this is it from the day 2. And if you want to ask any questions or any query, I guess I'm very much open. If you want to, if you have any confusion, you can come and talk to me, right? But tomorrow, actually, we will start with the cord blood stem cells. We will see. Anyway, so whatever the confusion or the problem you are having with this the 2 days lecture, you can discuss with me, I guess. So I guess thanks again, and then again we will start tomorrow at 3 o'clock. Okay, thank you.